Hi viewers, welcome to SEKHAR'S Bio Factory for NEET. Sexual reproduction in flowering plants with endosperm development. If you like this video, subscribe my channel and click on bell icon to get notifications. Like, follow and share my videos to your students and friends to get more information in biology related to NEET and other national state level competitive exams. In this video, I am going to explain endosperm development along with various important characters related to endosperm. A phenomenon called double fertilization. It is necessary for the formation of endosperm and embryo during sexual reproduction in plants. At the time of sexual reproduction, pollen grain it consists of two male gametes. These two male gametes reach the ovary, ovule, and embryo sac with the help of pollen tube. When the two male gametes are discharged into the embryo sac, at this time, one of the male gamete it fuses with egg cell to form diploid zygote. The another male gamete it reach the two polar nuclei of central cell, and after fertilization it becomes into a triploid primary endosperm nucleus. Similarly, it becomes into endosperm tissue. Endosperm formation it is generally pre-fertilization tissue in gymnospermic plants, but in angiosperms it is a post-fertilization tissue. The endosperm tissue, which is present inside the seed in most of the angiosperms, it appear after fertilization and then it provide nutrition to developing embryo. The formation of endosperm initially it takes place with the help of mitosis division. In this mitosis division, the nucleus of primary endosperm nucleus it undergoes endomitosis to become a triploid multicellular tissue. It is called endosperm. It is occur prior to zygote division. Generally, in most of the angiosperms, the primary endosperm nucleus it is a triploid in nature. The development of endosperm it takes place from primary endosperm nucleus, and at the time on the basis of first and other subsequent cell divisions which occur in primary endosperm nucleus for the formation of endosperm. This developmental process is basically three types. In this, the first type is nuclear type, the second type is cellular type, and the third type is helobial type. In nuclear endosperm development, the triploid primary endosperm nucleus it undergoes free nuclear divisions to form many nucleus. After completion of this free nuclear division, all nuclei present in central cell they push towards the peripheral side due to formation of a large central vacuole. Now, the cell wall formation takes place around the nuclei. It takes place from peripheral to center. That means the nuclei which are present at peripheral they can synthesize cell wall around it. Then the cell wall formation takes place towards center. Such type of endosperm development it is called nuclear type. The nuclear endosperm is the most common type of endosperm present in most of the angiosperms, and it is named based on initially at beginning it contains free nuclei. Example for this: maize, wheat, rice, sunflower are the best example for nuclear endosperm type. In most of the cases, the cell wall formation around the nuclei it is incomplete. Example, in coconut, the endosperm is multicellular. It is present at the outer side, that is peripheral region. Due to having a free nuclei in center, it become liquid. The young coconut, the endosperm, it is in liquid form with various number of nuclei. As the coconut grows, the number of cells grows initially and these cells are deposited at peripheral. Now it becomes into soft, tender and white kernel coconut that is we consume. The water which is present inside the coconut, it is basically liquid endosperm. The coconut water it is considered as energy drink because this coconut water is rich with minerals, vitamins, sugars, proteins and also main thing it is isotonic with our body fluids. This coconut water it is generally low in calories and contain various natural enzymes and minerals like potassium that makes it as a super drink. While it is good to sip on fresh coconut water any time of the day, drinking it at the right time can beneficially double health benefits that we can drive. The second type is cellular type. In this type of endosperm development, the primary endosperm nucleus it undergoes first division transversely that leads to the formation of cell wall in between these two nuclei. Now, after SEK2 cells, it divide into many divisions which makes 4 to 8, 8 to 16 every time cell wall formation is compulsory. Finally, many cells are formed inside the central cell that makes it is as cellular endosperm. Such type of development is called cellular endosperm type. Example for this, datura, petunia plants have this cellular type of endosperm development. The third type is halobial endosperm. It is a special type of endosperm. It shows both nuclear type and cellular type. In this development, the primary endosperm nucleus it forms two cells, in which one of the cell which is present towards microphylla region it is bigger in size and another one is smaller in size. The free nuclear cell divisions occur in bigger cell. It shows a nuclear type of endosperm development. After completion of nuclear division, all these nuclei are pushed towards peripheral region due to central vacuole. Simultaneously, the small cell which present towards chalazal end, it form 2 to 3 cells and it becomes into Hasturian cell which acts to attachment. Example for this, Asphodelus is a plant having halobial type of endosperm development. Endosperm it present around embryo to provide nutrition during development. It provide food materials in the form of starch. Sometimes it contains oils and proteins. The endosperm is starchy in rice seeds and endosperm is with oil in nature in castor seeds. Endosperms having proteinaceous structures and it is proteinaceous in nature in most of the cell. During the growth of endosperm, it crush the nucellus tissue present around embryo sac. Why? Because this embryo sac development it takes place by receiving food material from surrounding nucellus tissue. The endosperm after fertilization it is completely or partially eaten by growing embryo. After seed maturation, the endosperm may be present or completely consumed by developing embryo. Based on the presence or absence of endosperm in the structure of a seed. The seeds are basically two types. In this, the first type is albuminous seeds or endospermic seeds. The second type is X-albuminous seeds or non-endospermic seeds. In albuminous seeds or endospermic seeds, the endospermic seeds, those that have endosperm even after seed maturation. It is a fleshy, oily, surrounds the embryo and function as a sole food storage organ inside the seed coat. The embryo or cotyledon, it is present in the form of a thin or papery structure. In most of the monocotyledons and in some dicotyledon seeds, the food reserve remains in the endosperms. They are called endospermic seeds or albuminous seeds. In X-albuminous seeds or non-endospermic seeds, the seeds do not have any endosperm because the endosperm is completely consumed by developing embryo. Example for both endospermic and non-endospermic seeds. The dicotyledon X-albuminous seeds, example pea seeds, bean and gram seeds. Dicotyledon albuminous seeds, example castor and clustered apple. Example for monocotyledon X-albuminous seeds, Amorphophallus, Vallisneria, Alcima. Example for monocotyledon albuminous seeds, maize, wheat, barley, and etc. In wheat, the endosperm is grinded into flour for bread. The rest of the grain is included as well in whole wheat flour. Enriched wheat flour maybe contains a good source of irons, thiamine, niacin, calcium, and vitamin B6 in addition to above nutritions. The wheat have high nutrition value as compared to rice because it contains more protein and fibers than rice. The barley seeds having endosperm and it is the main source for beer production. The maize grain, it is called fruit because it is not a seed. During the development of fruit, the complete separate ripened ovary, it becomes into maize grain. In maize, a part of endosperm is sugary and another part are with starchy. It is found in two different color, some of the portions with white and some of the portions with yellow patches. So that such type of endosperm is called mosaic endosperm. The best example for mosaic endosperm, it is maize seed. Young maize seed, it secretes some of the milks. It includes cytokinins and very common type of cytokinin like geotin. It is extracted from milky endosperm of maize seeds. But matured endosperm, it never having such type of cytokinins and it never stimulating property. Rice seeds. In the structure of rice seed, endosperm, it occupies most of the portion, nearly 70% of the portion in seed, and it provides energy and materials for germinating seed at the time of seed germination. In addition to, endosperms serve as an important food source for human being since pericarp, seed coat and embryo, these are removed during milling and polishing before cooking. The aleurone layer is the outermost layer of endosperm followed by the inner starchy endosperm in cells. This layer of cells is referred as peripheral endosperm. The aleurone layer, it is also triploid in nature because it is derived from endosperm. The aleurone cells contain several aleurone grains which contain hydrolytic enzymes. It includes amylase, protease, etc. These are useful and helpful during seed germination. One most important type of endosperm, that is the ruminate endosperm. Ruminate endosperm, it is the condition in this the endosperm present in a seed, it is dissected by inner growths of seed coat. Ruminate endosperm, it is arise due to ingrowth or infolding of seed coat during seed development. Example for ruminate endosperm. It is found generally in nearly 32 of angiospermic families like Rubiaceae, Anonaceae, Aristolochiaceae, Pamaceae and Myristicaceae families. These are example for ruminate endosperm present in palm seeds. The layer of a nutritive tissue derived from nucellus that surrounds the embryo of seed in some angiosperms. Such type of tissue, it is called perisperm. The perisperm generally it is a diploid food storage tissue. It originates from nucellus tissue with the help of mitosis division. During the embryo sac development, the endosperm it receives food material from surrounding nucellus tissue. At this time, the nucellus is completely degenerates. But very rarely, the nucellus tissue it is still present even after seed maturation. Such type of remaining nucellus tissue which is left after the fertilization and absorption by the endosperm and embryo, that is known as perisperm. Some of the seeds which contains perisperm, the such type of seeds are called perispermic seeds. Example for perispermic seeds: sugar beet, coffee and black pepper. Endosperm in gymnospermic plants. In gymnosperms, there is no real endosperm because this nutritive tissue it is necessary for the growth of germination of embryo. It is formed before fertilization. Endosperm of gymnosperm, it is a free fertilization tissue. It is basically female gametophyte and it is haploid in nature compared to angiospermic plants. Some of the important terms related to endosperm. The first term is xenia. The direct effect of pollen on structures which present inside the embryo sac, that is except embryo, that is nothing but endosperm, this effect is called xenia. Example, in maize, due to xenia, different color grains are formed, that is called xenia effect. Metaxenia, the direct effect of pollen on the structures present outside of the embryo sac or endosperm, especially seed coat, fruit wall, is called metaxenia. Example for metaxenia: maturity time and size of fruits in date palm, it is example for metaxenia. Special features during endosperm development related to its ploidy. Endosperm is generally triploid because two polar nuclei and one male gametes are fused at the time of fertilization. In gymnosperms, endosperm is generally haploid in nature because it is a pre-fertilization structure. In Enothera type of embryo sac, the endosperm is diploid because only one polar nuclei is present in central cell. It fuses with one single male gamete. In Phenia, Plumvizella, Fritillaria, Flumbago type, the endosperm is pentaploidy because four polar nuclei are fertilized with one male gamete. In Peperomia, the endosperm is nonaploidy because the embryo sac contain 8 polar nuclei. These are fertilized with one male gamete. Highest number of ploidy, it is present in the endosperm of Arum maculatum plant, it having 24,576 ploidies in endosperm nucleus. Thank you for watching. If you like this video, subscribe my channel, like, share my videos to your students and friends.